Hi, good morning. Welcome. We're going to be talking about nucleus today. Nucleus, trying to give you as much of a complete understanding as possible about this function in the cell and this little organelle. That's important. So let's start from the beginning. What is a nucleus? Nucleus is a vital organelle in eukaryotic cells. It acts as a control center that regulates various cellular functions by managing gene expressions. So let's get into it. So today we're going to cover the structure, the function, nuclear dynamics, different cell types, diseases, research and application, and the potential evolutionary history, maybe even the future of the nucleus and what can happen. So like I said, here's a general cell. We will be delving into each of these as time goes, but for today we're going to focus on the top right, which is a nucleus. So you will talk more about them, but you see you have a nuclear envelope, you have a nucleolus, and you have a chromatin. So starting with the structure of the nucleus, as we know, we have the nuclear envelope, which we have the nuclear envelope, nucleoplasm, chromatin, and nucleolus as the four big pieces of structure for a nucleus, and things that we need to keep an eye out on, things that we should know about, how they impact, and what they really do. So without further ado, let's get started. So we're starting with the nuclear envelope. It's a double membrane that surrounds the nucleus. It separates the nucleus, surrounds the nucleus. It separates its contents from the cytoplasm, so it prevents all this DNA, all these proteins, RNAs, all that stuff from leaking out into the cytoplasm where it can mingle with all the other things. So in the nucleus, the nuclear envelope, you have three parts. You have an outer membrane, an inner membrane, and nuclear pores. The outer membrane is continuous with the endoplasmic reticulum. It may have ribosomes attached, so it has protein impact. The inner membrane faces the nuclear interior. It's lined with the nuclear lamina, which is a network of protein filaments that provide structural support. The nuclear pores, those are large protein complexes that penetrate the nuclear envelope. It allows the selective exchange of materials like RNA, proteins, etc. between the nucleus and the cytoplasm. Nucleoplasm. So the nucleoplasm, it's a gel-like substance within the nucleus where various nuclear components are suspended. Similar to cytoplasm, it contains water, nucleotides, enzymes, and ions, providing a medium for nuclear processes. So the nucleoplasm is kind of more where everything is kind of found and where all these materials kind of come together and start all these processes that happen in the nucleus, which we'll get into. So then you have the chromatin, which is the material that makes up chromosomes. It contains the genetic blueprint of a cell, basically. So there's a couple of parts of a chromatin that we need to know. So you have the DNA, which is the cell's genetic material. It's organized into linear molecules. Then you have the histones, which are proteins around which DNA is wound, helping to package it into a compact, organized structure. And then there's two other concepts. There's a euchromatin, which is less densely packed chromatin. It's typically active in transcription, and it's a key part of how we get DNA, how we get chromosomes, how we get all of these genetic material structured around how we are and who we are. And then there's the heterochromatin, which is more densely packed chromatin, usually is transcriptionally inactive. So euchromatin is the less densely packed, more impact, and heterochromatin is more densely packed, less active. Then you have the nucleolus. So the nucleolus is it's the site of ribosomal RNA synthesis and ribosome assembly. So this is kind of where the protein aspect of it starts to come. And it's a dense spherical structure within the nucleus that is composed of DNA, RNA, and proteins. The nucleolus organizes around specific chromosomal regions called nucleolar organizer regions. So now we're going to delve a little into the functions. So you have the genetic information storage, the gene expression and regulation, ribosome biogenesis, cell cycle regulation. So let's start with genetic information storage. So that's where the nucleus houses the cell's DNA, which contains the instructions for building and maintaining the organism. DNA is then organized into chromosomes, with each chromosome carrying numerous genes. Then you have gene expression regulation. So we have transcription. Nucleus is the site where DNA is transcribed into messenger RNA. You have transcription factors and other proteins then regulate which genes are turned on and off, which then creates a huge cascading reaction through the body, which is how you get sometimes all these diseases, how certain drugs work the way they do, by turning on and off genes, and aspects like that. That's where you get all these things cascading down and how things happen in the body the way they do, through gene expression and regulation. Then you have RNA processing, in which newly synthesized RNA is processed within the nucleus, including splicing, which is removing introns, capping and adding a poly A tail to mRNA before it's exported to the cytoplasm. So thirdly, now we're going to go to ribosome biogenesis. So here, the nucleolus within the cell is a site where ribosomal RNA is transcribed, processed, and combined with ribosomal proteins to form ribosome subunits. These subunits are then exported to the cytoplasm, where they assemble into functional ribosomes and actually start doing their work. So lastly, we have cell cycle regulation. So the nucleus is central to the regulation of the cell cycle. It ensures that the DNA replication occurs correctly during the S phase, and that the cell is ready for mitosis, otherwise known as cell division. The nucleus also contains checkpoints that monitor and control the progression of the cell cycle, ensuring that damaged DNA is repaired before the cell divides. This is especially important because damaged DNA is what causes all of these other issues. Cancer, carcinogenic, like carcinomas, melanomas, other types of autoimmune conditions, for example. All this stuff comes from cell cycle regulation, cancer being a big one, and these checkpoints are actually a key part of cancer therapy that determines how we target them, how we manage these conditions. Nuclear dynamics. So we have nuclear import and export. The nuclear envelope's pores control the movement of molecules in and out of the nucleus. Proteins, RNA, and other molecules are selectively transported through these pores via specific signals, like nuclear localization signals, NLS, and nuclear export signals, NES. More about that in a future video. Nuclear changes during cell division. So we have during mitosis, not before, but during mitosis, nuclear envelope disassembles, allowing chromosomes to be separated into daughter cells. After mitosis, the nuclear envelope reassembles around the newly formed nuclei. Alright, so you can see where the nuclear changes come in. During mitosis, the nuclear envelope breaks so the chromosomes can split. And after mitosis, it comes back together, so you have a new nuclei now. Now we have two different types of cells that have nucleus. We have multiple nucleated cells, so they have multiple nuclei. Those are usually found in muscle fibers and certain types of fungi. And then we have atypical nuclei, in which red blood cells in mammals lose their nucleus during development to maximize space for hemoglobin. So like I said, those are usually found in red blood cells, and they take up less space in the cell, so you can fit more of what we need. So now moving on to diseases related to nuclear function, so we have cancer. Cancer is the biggest one. So we have mutations in nuclear DNA lead to uncontrolled cell division and cancer. The nucleus is also involved in response to DNA damage, which, if faulty, can contribute to carcinogenesis. And like I mentioned before, targeting these mutations and preventing them, so monitoring the nucleus, monitoring everything that comes out of the nucleus, comes with the nucleus, is key to preventing and treating cancer things, which is why when you see medications and you wonder how they work for cancer, they work by targeting mutations, by targeting the checkpoints, by regulating those, and that's how we know. Genetic disorders, which is second, so we have mutations in nuclear DNA or in the proteins that regulate nuclear processes, can lead to genetic diseases such as Huntington's disease or cystic fibrosis. And those are important because while these are rare, they do exist, and it gives us a better understanding. If we understand how the cells work, if we understand how the mutation in the cell in the body creates what it does and why we have these disorders, it's easier for us to determine a cure for it. A cure, not a cure, but a treatment. Last but not least, we have nuclear envelope diseases. So we have defects in nuclear envelope proteins, such as laminopathies, can lead to diseases like progeria, which is premature aging, basically. So research and applications. So we're looking at things like nuclear transfer. We have somatic cell nuclear transfers, SCNTs, which are used in cloning, where the nucleus from a donor cell is transferred into an egg cell whose nucleus has been removed. This allows us to maybe mess around with cloning. There was a famous study with a lamb that was done with it. And then there's gene therapy. The nucleus is the target for gene therapy, where corrected genes are introduced into the cell to replace faulty ones. Now that's a big one. If you can just straight up yank genes from person A and put them into person B to replace faulty ones, that can be big for our medical field, for our big medical and scientific fields, because that's a key thing. We can basically transfer, we can change a lot of stuff like that. And that's something that they're looking into for the future, and actually might be something to keep an eye on if that's what interests you. So now looking at things evolutionary-wise, we have the endosymbiotic theory. Now the endosymbiotic theory primarily tells you how the mitochondria and the endoplasmic reticulum came to be, but it does have an impact on the nucleus because all of those came as a result of ancient prokaryotic cells. Now while the nucleus primarily identifies eukaryotic cell in eukaryotic cells, because prokaryotes don't have nucleuses. This is important because it came as a result of evolution through a symbiotic relationship between the ancient prokaryotic cells, which eventually gave rise to the eukaryotic cells. And that's in here you have compartmentalization of genetic material, because like I said, you have the double membrane that protects the material from flowing out of the nucleus. And that's a key thing, and that provides advantages in regulating complex cellular processes. That's all that we have on the endosymbiotic theory for nucleus, but there is a lot of prospect and a lot of future in learning and studying the nucleus. We've already done most of it, but as we go as more diseases come up, as we make scientific advancements, we can really identify that there's a lot more that we still have to learn about the nucleus and its effects on diseases, its effects on our treatments, our existing things, our body overall. And that's something that we can really look into and where our future really holds. And that's all I have for you today. I know that was just a very basic one. If you have any questions or concerns, leave them in the comments. If you have any topics you want me to cover, also leave them in the comments, and I'll do my best to cover them in any future video. Thank you very much. Have a wonderful day.